machine here is called a centrifuge. In the lab, we use it to spin blood samples at a very high speed so that different components of blood can separate. That is, the heavy components will move to the bottom while the lighter ones will move to the top. Next is an autoclave. An autoclave is a sterilization machine and it uses steam under pressure at usually 121 degrees at 15 psi to kill microorganisms. So it sterilizes culture media and laboratory instruments. Next is the hematocrit centrifuge. This is a special centrifuge that is used to spin capillary tube for packed cell volume tests. This instrument is known as micropipette. It is a precision instrument that is used to measure and transfer very small volume of liquid, and it measures liquid in microliters, and it comes in different sizes. We have the 20 microliter, the 1000 microliter, and the 200 microliter. Next is the laboratory refrigerator. It stores samples and reagents at low temperature, usually between 2 to 8 degrees centigrade. So it is used for storing reagents and preserving blood samples for future use. This machine here is known as hematology analyzer. It is an automated machine used to analyze blood cells. So it is used in the laboratory to carry out full blood count tests, white blood cell counts, and hemoglobin measurements. Next is the blood mixer. It gently mixes blood samples with anticoagulants to ensure proper mixing, therefore preventing clot formation. Next is the sharp box. This box is used to dispose of sharp medical objects like needles, blade, and lancets. Next is the microscope. The microscope is used to magnify very small cells like red blood cells, white blood cells, platelets, and it's also used to magnify microorganisms such as parasites, fungi, and bacteria for identification purposes. Bunsen burner. Bunsen burner is a laboratory gas burner that produces a single open flame. It is used to sterilize inoculating loops for culture and also for heating of substances. Electrophoresis machine. This machine is used to separate molecules using electric current like DNA, RNA, or proteins. So in the laboratory, it can be used to carry out genotype tests. Next is the water bath. The water bath keeps sample at a constant temperature using heated water. It is used for warming reagents, incubating samples, and for enzymatic reactions. Next is the incubator. It maintains a constant temperature, usually 35 to 37 degrees Celsius, and it is suitable for microbial growth. In the laboratory, it provides a suitable temperature needed for the growth of bacteria and fungi. Hot air oven. Hot air oven is the sterilization equipment that uses dry heat. It uses dry heat to sterilize and dry glassware and microbiological plates used for culture.